Hey everyone, today we're going to talk about life is cellular. So some of our key questions: what is cell theory? How do microscopes work? And how are prokaryotic and eukaryotic cells different? Vocabulary you will need to define: cell, cell theory, cell membrane, nucleus, eukaryote, and prokaryote. And so in your book you should outline before you read. Make an outline using the green and blue headings in the text, and as you read, fill in notes under each heading. So those are just bolded, they're large and in charge, so even if you're colorblind, you can do it. Just look for the bolded words, make an outline, and fill it in. Alright, so we're going to start with the discovery of the cell. And in the 1500s, eyeglass makers in Europe discovered that using several glass lenses in combination could magnify even the smallest objects to make them easy to see. And in about 1665, Robert Hooke came up with the term cell while looking at a slice of cork. And he said that he could see things inside that kind of reminded him of tiny rooms, based off of a monastery tiny room, and those were called cells. And so he used the term cell because they looked empty to him, but today we know that they are not empty and they have a bunch of things inside of them. Around the same time, Anton van Leeuwenhoek used a single lens microscope to view pond water, and this figure over here is what he saw in the water. And so he took other samples, actually organisms that we can find in the mouth, like bacteria. Actually that's what that figure is, my bad. These are bacteria from the human mouth, but he did look at pond water as well. Alright, and so based on everybody's research and things that they have discovered, we developed the cell theory, and there are three main components to the cell theory. All living things are made up of cells. Cells are the basic units of structure and function in living things, and new cells are produced from existing cells. Later we'll get into cell division, mitosis and meiosis, and how they actually do that. Alright, so let's talk about microscopes. Microscopes use lenses to magnify an image by focusing light or electrons. So here we've got a light microscope. This is actually onion cells. So light microscopes use light to view an image that has been stained or dyed. So without the stain, they're translucent and you can see through them. So the stains help to highlight different areas, and different parts of the cell can be dyed different colors as well. What else? You can view live specimens, underneath a light microscope, so that's an advantage. But electron microscopes allow you to zoom in much more. So these are all yeast cells viewed under different types of microscopes. So this would be a light microscope, and then these are two different types of electron microscopes: transmission and scanning microscopes. And these use what we call false color. We use a computer program to add the color later. So I like to call this, like some fancy computer editing program place. Maybe you've heard of them, Pixar. I like to compare this to the Pixar type of microscope, where you go back later and you add the color in the computer image. Alright, downside to this, these samples are not alive. These samples need to be sliced very thin in order for you to see through them. But you can view things like DNA and viruses. Both of these can be used to view prokaryotic and eukaryotic cells. So let's take a look at those. Eukaryotic cells have organelles. So anytime you hear the word organelle, we are talking about eukaryotic cells. And that's one of the main differences between eukaryotic and prokaryotic cells, is that eukaryotic cells have organelles, and we keep our DNA inside the nucleus, which is a membrane-bound structure. Prokaryotes do not have a nucleus. They have a nucleoid, which is called a false nucleus. And inside the nucleus, we have, just pretend this is my nucleus, and we've got some DNA in here. It's just hanging out, and right now it's called chromatin. And then inside we also have a nucleolus, which is where ribosomes are made. And when our cell's getting ready to replicate and divide, our chromatin will wind up tight into chromosomes, and then this is what we're normally talking about right before cell division. So we've got our chromatin and our nucleolus. Chromatin has our DNA. All of that is inside of the nucleus. So that's what this nucleolus is right inside there. All right. Differences. Eukaryotic cells, they have organelles. We keep our DNA in the nucleus. What else? Prokaryotic cells have a cell wall. Eukaryotic cells, this is an animal cell. So this one does not have a cell wall, but plant cells do have a cell wall. And all cells have a cell membrane. So all cells have a cell membrane, and they have genetic information in them. And then another thing they have in common is ribosomes. Ribosomes are used to make protein, and then proteins are used to carry out daily functions of the cell. And they also determine our traits. So proteins help determine our traits. Some examples of prokaryotes would be bacteria and archaea. Examples of eukaryotic cells would be protists, which is like red algae, fungi, mushrooms, animals, you and me, kitty cats, whatever, and then plants. Those are examples of eukaryotic cells. All right, so let's review. The cell theory states that all living things are made up of cells. Cells are the basic units of structure and function in living things. New cells are produced from existing cells. Microscopes use lenses to magnify the image of an object by focusing light or electrons. And then the main difference between prokaryotic and eukaryotic cells is that in eukaryotic cells, the nucleus separates the genetic material from the rest of the cell. So eukaryotic cells store their genetic material within a nucleus, and prokaryotic cells do not separate their genetic material within a nucleus. All right, guys, I hope that made sense. See you next time. Bye.